let's get started from where we left. And where we left was that we were thinking about how can we use this, confocal imaging and really quantitative approaches to understand cell shape, to understand this process of changes in shape of the early embryo. And so I'm now going to show you a couple of things that we have done with the C-star embryos that are showcasing this and how we found some variation in this process and how we're thinking about describing and understanding this variation. And so these are stills from one of the movies that I showed you before. And at these stages, the C-star staging is simply the number of cells that are in the embryo at that particular stage. So it's a cleavage thing. And the kind of information we can get is, but cell by cell, we can, for instance, look at the cell volumes. And as you see here, as we expect at each cleavage, the cell volume halves, right? And that's exactly what we knew would happen. And then we can get tissue level properties like cell density. And we see that cell density doubles at every cell division, because obviously there is about double the amount of cells for more or less the same space. And then we can do things like we can quantify other aspects of the dynamics of this tissue. And for instance, here we quantified the area of these openings that I showed you before that are getting smaller and smaller and then eventually close, right? And so we get a quantitative description of this phenomenon. And we know that for most embryos, this closure happens at around the 512 cell stage, right? But if you pay attention here, there is massive amounts of variation already there in that data set, right? And so basically what seems to be,
That jump, like a sign of like synchronized division.
Yeah, so if you, let me see, where's the earliest movie that I have of this? Here, right? So we have looked at this here, and basically every time that there is a cell division, the size of these openings are kind of jumping, because what's happening is that the cells are kind of, because of the oriented cell division, they pop next to each other. So you are thinning the tissue and spreading it, right? And it seems that at every round of cell division, there is a jump, right?
The lumen is moving at a different rate than the surface.
Let me show you this. I think this is going to be... No, don't worry. So this is a cross section. The time resolution is less than what's before, but it shows you what happens is that the cells are organizing on the surface. The surface is the fertilization envelope, right? So they are organizing on the inner surface of this fertilization envelope, and at every round of cell division, they kind of pop on the side. And so that's why I'm saying that it seems like the tissue is thinning and spreading and covering all the inner surface of this envelope.
Tight junctions everywhere?
Unclear. There is a polarity, there's junctions. I don't know if there's tight junctions at these stages already. Eventually, when it closes, there is an inside and an out and flow control. All right, so what seems to be happening is that the cells are kind of occupying all the space that is available to them. And so one obvious question was like, what if they had less space, right? You would predict that they would change the dynamics of this closure. And so in order to answer that question, we did this simple experiment that we took the zygote and we put it in a gel. And what that does is that the envelope sort of collapses onto the zygote, and then effectively it gives the cells of the embryo less space to develop into, right? And so we can do the same sort of movies and the same sort of quantification. And what happens is that these are your normal embryos and these are your compressed embryos. And as you can see, development occurs more or less at the same time. So the cell divisions keep going, development progresses, but it
control the stiffness of the environment.
Basically, yes.
Can you increase the stiffness?
We haven't done that. Yeah, we haven't done that. I just found a level. So there is a threshold. If it's too soft, you're not going to do anything. If it's too stiff, they're going to die. So we're kind of in the middle of there where we can compress them without destroying them. And so cell volume is not changed. So here in these graphs, you're seeing stage by stage, you compare wild type and compressed, right? So there's no difference in the cell volumes, but cell density at each stage is actually increased. So we are actually compressing them. And as you might have seen already from the picture, these openings are closed a lot earlier, right? We can anticipate the closure of the epithelium. And this is kind of important because, as we were kind of hinting at, you need to have a closed epithelium in order to control your inside and your from the outside, right? So if you want to build pressure, if you want to have a different environment, and then being able to go do the next step, which is gastrulation, you need to have an epithelium closed. And the timing of that with respect to the cell cycle can be changed by changing the space that is available, which is kind of a big thing. And so closure, timing, the cell density depend on available space. But the past day we were talking a little bit about topology of cells, so I thought I mentioned this. Have you ever heard of a scutoid? Right? Okay. I had not heard of a scutoid before I met Luisma at a conference, and that's his whole thing. They found this shape that cells acquire in curved epithelia, and his work and the work of Timothy Sanders describe this new shape. And so when I met him, they were working on the Drosophila salivary gland, and they were mostly doing stuff on fixed samples, and he was complaining that they didn't have a system to look at the dynamics. So we have this beautiful curved epithelium. Why don't we look at the scutoids in there? And he told me it's a sphere. You're not going to find any because just for those of you who have not heard of a scutoid, what this is like if you normally think about a monolayered epithelium and the shape of cells within that epithelium, the majority of cells will have a prism or a frustum shape, which means the neighbors that they have on the top, on their apical side, are the same neighbors that they have on the bottom, on their basal side, right? But in some epithelia, especially when they're curved and when there is anisotropies of curvatures, you'll find a proportion of cells that acquire this scutoidal shape, which is nothing else than an apical-basal transition. So it means that you have a different set of neighbors on your apical side than you have on your basal side, right? This is one way to recognize them. So you see this yellow cell here is neighboring red, blue, and green, but on its apical side, but on its basal side, it's only neighboring red and green. So kind of those cells have snuck in the middle of the contact and have eliminated the other neighbors. So that creates this shape, which is a scutoid, right? And the theory is, which, Luisma and Timothy don't entirely agree on what the theory is, but the consensus is that they form more often when you have curved epithelium like a tube because of the anisotropy of curvatures in the two directions, creating anisotropies of tension, and then the shape of the scutoid allows the cells to cope with that and keep the epithelium closed, right? And so what we find, in principle, in a sphere like the sea stars, you shouldn't have any because there is no anisotropy of curvature, right? And then Luisma told me, we're not going to find any. We cannot use this system. And I said, why don't you just take this movie and just look. And what turns out is that there are no scutoids at early stages, but once the epithelium starts closing and becoming more compact, they appear. Right? Some of the cells do acquire this shape. And when we compress the embryos, a higher proportion of cells takes up the shape of a scutoid. So this is changing the topology of cell-cell contacts within the epithelium, not just the cell density. And to make a long story short, there shouldn't be any scutoids because of the shape of the embryo, but there are these rounds of cell division that create probably imbalances in the forces between apical and basal sides. And what we found, what Antonio found, Antonio tracked about 200 of the scutoid cells in the embryo, and he found that they most often form right after those cells have divided. So when these rounds of cell division are happening, you get many of the scutoids that then kind of get resolved over time. But then there's another round of cell divisions and they form again and then they get resolved.
Do they have also a biological implication?
We do not know. But yeah, so the most obvious biological implication is that you're changing the set of neighboring cells, right?
It's downstream of that.
We don't know yet. Not in this system.
So how long are these, how stable are these?
They can last for hours.
So it's
Yeah.
not like it's got between a T1 transition. Sort of.
So it is a very slow, it can be a very slow transition. So we found the majority of these scutoids last for the whole cell cycle and then they get resolved just at the end. So this can last for an hour, right? And then the cells divides and it gets destroyed. And so what it seems to be doing is that it starts from one side and then it kind of elongate until it's resolved. It's this, it looks very much like a T1 transition in the other axis, right? Yeah, but it's very different in different systems. They have now been observed in many different epithelia, and there is some work on, I think the group is Kazza. I'm not sure I'm pronouncing their name correctly, but they have seen it also in Drosophila, and they've seen some of them are very long-lived, and in some other situations, instead, they are not. So this is sort of a newish kind of set of things that have been, we are just starting to describe them. Yeah.
Is it true that maybe under the extreme compression confinement condition, you prolong the culture and the epithelial cells will develop from a monolayer to a multi-layer, and then this scutoid observation, it maybe is provide an onset for that.
Yeah. So what happens when we compress them is that at some point the tissue folds inside. So I was hoping to make like a really tight early cells. It doesn't work that way. They stay on the surface, and then when there is no more space, they fold in. And so the curvature there also can be, it's changing then. In order to get a sense of what is the role of compression alone, we only analyzed areas of the epithelium where there wasn't a fold. But yeah, and how that affects cell differentiation is something we are looking into right now, basically.
From the engineering perspective, I'm wondering whether it's possible, technically possible, you can modulate the luminal pressure.
Yeah.
I think it's doable. Assuming it's doable.
I think it's doable. We haven't done it yet. You can change the osmotic pressure around, and you're going to change the osmotic pressure inside. But these embryos are osmoconforming, so when you cannot. They do okay with a range of salinities, but you are changing things. I haven't had time to do it, but I really want to try to just put a needle inside.
Same.
Yes, you can definitely do that. You can put a needle inside and open it up, but I haven't done it systematically yet.
Can we seal it on the needle?
I'm not sure yet. Yeah, I'm not sure either.
We have done that for stem cell-based models.
Okay, cool. Yeah. You had a question?
Okay, in the compressed embryos, do you see any alterations with cell division orientation? Do you get orientation perpendicular to the surface?
So to be entirely honest, we haven't really quantified that precisely. Just by looking, not really. It seems like mostly they kind of keep dividing that side and then they fold. But it is something that we are doing now with a slightly different setup that I'm gonna talk about right now. Because I was not particularly happy with the compression in the gel, right? Because the point is, okay, we found that now closure timing, cell density, and topology depend on the space that is available to the embryos, right? Who cares, right? You compress something, it's gonna do something. But the interesting aspect of this is that normally in development, right, in nature, the space that is available to the cells is set by the size of the fertilization envelope, right? By the diameter of the fertilization envelope. And so what is also interesting is that we can do now these movies where we image tons of embryos, right? This is another thing that is great about this model systems is that you get millions and millions of eggs every time you do an experiment. And now, as microscopes get faster and faster, this is simply a confocal image, and it's a tiling of many different, it's a tiling of, I think, 15 or 20 tiles that Sarine did while she was in the lab last year. And we can get an understanding of what is the variation of this dynamics of closure of epithelium and the variation in shape. And so what you can do here is that you can look at, for instance, when in time the epithelium is closed, which from our compression experiment, we know it's a proxy of cell density, right? So the more compact more dense, and earlier there, all of these openings are going to close. And so we see that there are some embryos in which it closes at 512 cell stage. There are some embryos in which it closes like 2 cell stages before, right? So there is some variation there. And when we go and, yeah, I already said that. So what we can do with these movies is now we can look embryo by embryo. We can measure the area of the zygote, the area of the envelope. We did this just measuring the area of the 2D cross section. And then we can look at the very same embryo, and we can record at which point in time during development the epithelium is closed, right? And when we do this for many wild-type embryos, we find this beautiful correlation by which embryos that have relatively bigger fertilization envelopes compared to the size of the egg also tend to close later, and embryos that have smaller envelopes close earlier, right? So this thing that we found experimentally by compressing the embryos in the gel might actually have something, might be relevant to the natural variation of early embryo development in the sea star. And so, as I mentioned, the space is set by the fertilization envelope. When is this fertilization envelope formed? In these embryos, it happens actually at fertilization, which is why it's called fertilization envelope. And you can do this fertilization in a dish, right? Very controlled. Put them in seawater, egg and sperm together, and there is a massive cortical exocytosis of cortical granules that happens at fertilization, and the material that is in the cortical granules gets pushed to the surface. It combines with proteins that were on the surface already, and they were making the vitelline membrane, and creates this envelope, right? That kind of lifts off the membrane. So it turns out that if we change the calcium and magnesium concentration in the seawater, we can change the shape of these envelopes. And so if we put embryos in seawater with very high concentration of calcium and magnesium, we end up with tiny envelopes, right? And we can now do our experiments of imaging tons of these embryos that are controls and with tiny envelopes, and we can quantify the closure. Yeah.
You need both calcium and magnesium?
We have done with both because
they're both divalent cations, and the size is not that different. So why would you need both?
Say it again.
They're both divalent.
Yes. It's possible that you just need one. Yes, it's possible. We haven't tested all the different media possibilities.
And Vanessa, I think often so calcium and magnesium will presumably go through and just generally raise the osmotic.
I'm not sure what is it that they do. Yeah.
So a very standard approach to control this, and we were just talking about, is to just add phycol.
Yes.
And the phycol doesn't get in, but it essentially dehydrates, so it compresses the envelope.
Yes. So I was trying to find a way where I don't have to keep them in a weird medium, because we do this for 2 minutes at fertilization, then we put them back in normal seawater. And it stays, right? So I think the ion concentration is actually doing something to change the enzymatic reaction that happen at fertilization. Like there is an ovoperoxidase that cross-links proteins. There are proteases that do another job.
That said, phycol on the outside of the vitelline membrane is probably quite harmless.
Possible. We could try that. Yeah, we could try that too.
You ever try monovalent?
We have not. This is all very new stuff. So
use potassium or sodium, would it change everything?
Not sure. It's a good thing to try. All of these salts are in seawater, so I'm assuming. Yeah, exactly.
Sodium is much more relevant than
perhaps. Yeah. It's a good question. We could try all different concentrations and see what happens. I think there's an entire project on how this fertilization envelope is changed by environmental conditions. Yeah.
Divalent cations can essentially bind and polyelectrolyte gels, they'll essentially serve as transient cross-linkers. But monovalent cations
will not do that. Oh, so that would give us an idea of the mechanism, right? Yeah, that would be awesome.
Usually charged, usually they're negatively charged, and so the cations can essentially serve as transient cross-linkers. I was wondering whether or not you're
possibly
with that.
Yeah, we haven't had the manpower to go into that yet, but I think that's an interesting question. I'm hoping we will have some time to figure it out. Yeah, I think there is that. There is also the possibility that the different ion concentrations somehow affect the exocytosis process. that it's what, push the material. So those are things that might be happening, but we don't really know how this works. We're just using it as a cool method. It's transient, and then the embryos are fine, and we put them back in regular water, and we can do this, right? And then if we remove calcium and magnesium from the seawater for these 2 minutes of fertilization, we get large envelopes, which is kind of cool. And so we can do the same experiment. Yeah. Yes, yeah. It's 10 minutes.
So this could be then hydrogen swelling induced.
Yeah. So the way there is two ideas, like the semi-accepted idea of how this fertilization envelope lifting works is that there's a bunch of mucopolysaccharides and other material that gets pushed in that space, and then that draws water and it lifts, right? So we can also change the relative size of the envelope. If we put the embryos in higher salinity water, the envelope will be smaller, will shrink. In that case, you have to keep it in the higher salinity water. If you put it back in lower salinity water, the envelope will lift more. So there is definitely an hydrostatic pressure type of, or osmotic pressure type of situation there to have the envelope lifting. Yeah? Okay.
We know different envelopes with different sizes. Have you measured the mechanical properties?
It's the next thing on the to-do list. Yeah.
And also from this video, seems like under cultivation, you have more failure modes.
You see a graph of that. Yeah. Yeah, so first quantification that comes out is that if we actually experimentally change the size of the envelope, we can either anticipate or delay this disclosure, right? So this is definitely, I put it, I like to plot it here because it's like an extension of the natural variation, but this is actually experimental setup. So there is significantly different in the two sides, right? So we have the size of the envelope and the space available to cells is definitely a determinant of dynamics of early embryo shape in these species, right? And so this is kind of, the idea would be that by changing the boundary condition, even if you don't change any other properties of the cells, you can vary the dynamics of this process. But probably this is not everything, right? And one of the reasons why I'm saying this is that if you look at this natural variation, there is also, there is some variation that is explained by the differences in envelope sizes, but there's obviously variation that is not explained by that, right? And so we have a situation where we probably have multiple things that vary and contribute to variation in the early shape and in these dynamics. And so what can we do to access that? And so I'm just going to tell you about a couple of methods that are available to us that we can use to try and see if this framework of surface tensions that have been used in the mouse and human embryo can be helpful to us to describe this variation and how. And so one of the things that you can do, for instance, is that you can, we have all this imaging, we can definitely measure angles, no problem. We can measure surface tension to the medium with the same assay that was used on the mouse embryo and the human embryo, which is this micropipette aspiration assay. And so what you do here is that you go in next to a cell with a pipette and that you have hooked up with a pump that is very precise so that you can just suck in a little bit very gently and slowly. You get a deformation of the cell into the pipette, and then you use Young Laplace to measure the surface tension. Cytoskeleton also. So you really measure cortical tension.
And do that happen simultaneously, or does the membrane come first and there's a blend?
If you do the assay right, they come together slowly. Exactly. Exactly. And then you're not measuring the effective surface tension anymore. You're measuring detachment of membrane from the cortex. So you have to do this relatively slowly. I mean, slowly, it's still just 2 minutes, right? So it's a fast measurement and you can repeat it, but it needs to be done properly. Otherwise, you have confounding factors. Yeah.
You only try this at the parcel stage?
No. So the cool thing is that you can then release it, the embryo continues developing, and you can do multiple measurements. So Jana in the lab, who is doing these experiments, has started measuring this at all different stages to understand how this property of surface tension of the cell is changing. You know, if you think about an average C-star embryo, what's the dynamic of cell surface tension over the period of time that this embryo shape variation that we are observing, this embryo shape variation, right? This is very preliminary, so don't quote me on this, but it seems like there could be a lowering of the surface tension over time and as the cells get compacted. But again, here you have a ton of variation, right? So we are trying to figure out where the variation is coming from. We have some ideas and figuring out what's the best way to use this tool to get an understanding of the physical properties of cells. And then variation between
the cells divide, right? But you can imagine that the total surface area is increasing, maybe like dilute the cortical.
Let me show you.
Possibly as one explanation.
Yes, exactly. So that is actually the most likely explanation, and I am gonna try to do this. So that's exactly what we find, that there is incredible changes in the surface tension as the cells are dividing. Right, so this is the same embryo that now Jana has measured over and over again during the course of the first cell division, whoop, and then second cell division, whoop, right? So this is a range of, it's a massive range, right? So there is an incredible variation of this parameter that she measured here at the polar sides of the cell division. And so obviously the question now becomes like, how do we do these measurements embryo by embryo to figure out if the variation we have is true variation or if it's just a timing variation, because these changes are so big. So now we are redoing this experiment following each embryo over the cell divisions, and obviously it's a little bit more difficult. Yeah.
It's the direction the cell divides into correlate differences.
So for now, we just measure. She just measured it at the equator, right? Equator? No, at the pole. Thanks. Thank you. She just measured it at the pole, and at each cell division, she kept measuring the pole because that's the easiest way. We are thinking about ways to measure in other points, but it's a little bit more difficult because the curvature then is not that reliable, and you have to kind of. So the pole is an easy hallmark to find, and you know that you're measuring always in the same place, cell by cell. Let's say you want to measure next to the cytokinesis ring. You need to know where the cytokinesis ring is going to form in order to do that, and that's a little bit more tricky, technically speaking. But yeah, those are good questions. Yeah.
Do C-star embryos have any kind of ECM?
No.
Similar to urchin?
Nope, they don't. Yeah, that's why I like them more. Yeah. So there's nothing around them. Okay, where were we? All right, we were here, right? Okay, so this is just, yeah.
So you see the decrease in the cell media cortical tension over time, but you see like the increase in compaction. So do you think at all that there's also yet further increase in the cell surface tension, or do you think it's really just driven by getting to that sort of boundary that's sort of forcing all the negative space to go away?
Yep. So here we go. So this is a quantification of the angles for the same cells that Jana measured the cortical tension of, right? And so that's exactly what you're saying. The cortical tension is going down, but the compaction is going up, angles are going up. So what's happening? If we think about this in the same framework that Jean-Léon and Hervé have used for their modeling of embryo and mouse and human embryos, this would mean that the tension, the interfacial tension between the cells is also decreasing, and it is decreasing faster than the cortical tension. And now then the question is that because cell adhesion is increasing, or is that because cortical tension at that side is decreasing? We are doing some imaging to try to figure that out. But there are also other experiments that you can do to measure directly the cell adhesion force, right? So you can separate the two cells with set force, and then you know what the force they were adhering with. And you can look at how, when you separate two cells, If there is a bulge, right, that tells you that there is lower tension at the cortex in that area compared to the outside of the cell. So we have tricks to sort of measure all of these parameters and see which ones are changing. Ongoing. You're welcome. Okay, and then basically this is what I wanted to say here, right? So we can measure these things in these embryos. The question is, how can we measure variation of these parameters, right?
There's an assumption there, which is.
Yes, there's many assumptions there.
Constant. That those interfacial tensions are constant. And why should that be true? Because as these adhesions start to mature, my suspicion would be that all those interfacial forces are changing. They start out relatively slow, and then they start becoming larger. Yeah. Can you actually observe changes in the angle and infer? So rather than fit it, can you essentially infer what was, because there are 3 parameters, right?
Yes, I think we can totally do that. Yeah. So the assumption is that the system is at equilibrium at every time point that you're measuring, basically, so that it reaches equilibrium faster than it's changing. And I'm not sure that this is true.
Theta, when you measure the theta, do you see a dynamical change wherein it starts out at one value and eventually equilibrates to a different one?
During the time of the measurement, right? So Yana is measuring the theta at in the same movies that she's using to measure the cortical tension, right? And during the time of that measurement, these 2 minutes, there isn't a big change.
What about in the early stage?
If you would follow it over the time of a cell division, which is 30 minutes, you would see the changes. And the cells round up as I think you can actually see. We haven't really done this quantification yet, but you can sort of see it if you do we actually have a video? I don't think we don't have a good video of this. Maybe this is the easiest way to show it. Yeah, anyways, you can definitely do that. We haven't done that yet. I was just talking with Jan a couple of days ago that this massive variation in cortical tension during the cell cycle give us a handle on how shape is changing, evolving, depending on that. Is that really changing the shape all that much or not? So we should use that cell cycle variation to get an understanding of how much this cortical tension is influencing the shape. It is somehow, right? Because you do see the cells rounding up more, the angles get smaller, and then they get a little bit bigger, but not so dramatically. All right.
So,
yeah.
Just a question on something similar. So even though it's a cleavage division, you will still have this sort of cell becomes very asymmetrical just before the division because of before it forms a ring and divides into 2. So if it's based on in what axis is the division going to be, it can cause both things, right? It can compress and basically reduce the angle, but if it's if it's dividing in a direction that is orthogonal to where you're measuring, it could be like, relaxing the angle as well.
Yeah. We are assuming, like this model assumes that the cortical tension that you measure at the pole is representative of the surface outside, right? Of the whole cortical tension outside of the contact, right? This may be true, it may not be true. Especially during the cell division, it's definitely not true, right? Because as the actomyosin skeleton is making the cytokinesis ring, obviously the tensions there are different than at the pole, right? So a corollary of these experiments is that we really need to figure out how to map cortical tension around. My hope was that if we do the measurement right in between the cell divisions, we would get some idea of how the steady state of the shape is now controlled. That was my hope. I'm not sure it's going to work out. We might become a more complex picture of this, but yeah. Other questions?
So the envelope itself is passive, you were saying, right?
Not sure. We'll see.
Okay.
I'll show you. Yeah, I mean, it's passive in the sense that it's proteins, right?
Okay,
It's a material,
sure, yeah.
and it's not doing anything, but the cells are interacting with it.
So there are interactions.
Say
There
that
are
again?
biochemical interactions between the dividing cells.
There are definitely physical interactions, right? So if you look at this
We know whether biochemical interactions,
chemical, I don't know,
biochemical, say,
Yeah,
adhesive
yeah.
interactions, we know.
we don't know.
We don't know. Okay.
We don't know. I can tell you that if we look at this video that I showed before here again,
You see it almost like finger-like structure.
exactly, yeah. So this is a cell mask. So this is just the red is marking lipids, right? So it's marking all of the membrane. You see how there are all these tethers that are remaining attached to the envelope, right? And now we have done some experiments. We know that at least in some of these tethers, there's actin, and there are like big patches of actin right under the envelope. And in some cases, I think you can see it a little bit here. In some cases, it's much more obvious. There is a divot in the envelope, in the point where the tether is. So there's definitely pulling forces. And now we are trying to figure out like how much is this pulling force? Is it actually enough that it could simply mean that the cells are adhering to the envelope, and then every time there is a cell division, they get pulled up, and that's how the envelope is controlling the shape, right? So I need to find a good way to measure the average adhesion force between a cell and the envelope, which is a little bit different than measuring cell to cell, right? So where were we? Okay, we were here. So these are sort of open points, things to think about when we try to approach this sort of questions, you know, like what is the actual variation of shapes that you can get in a specific species and in more species? How can we quantify the shape variation in an effective way and that is relevant to the particular problem that we are looking at and trying to understand? All of these biophysical measurements are super cool. Can we really use them to compare embryo to embryo? Are they sensitive enough to allow us to distinguish between noise and actual variation? Right. So those are things that we are thinking about. I'm not sure I have answers, but it could be good things to discuss about.
Can I ask you to unpack that first? You know, what would you like in your space?
So there's no picture of the pig. So I think these are examples that kind of exemplify what I'm thinking about when I'm thinking about morphospace, right? Even if you have a very simple set, like these 4 cells with an envelope, there is a certain range of shapes that you can get, right? So the C star, I think, is the simplest one. It's the 4 cells in their envelope, period. But then there are things like the sea cucumber, which obviously has a weird relationship with its envelope and between the cells, and it's changing but you can think of it as a modulation. You can think of a way to transform a C star shape to the sea cucumber by changing certain parameters. So this is what I'm thinking.
And you can do that. You can essentially.
I think somebody that has better modeling experience than me probably can.
No,
like I can see
an experimental perspective by perturbing.
Right. Yeah. Right. So the example of the envelope size is one parameter that you can perturb. Right. You can perturb cortical tension. You can perturb cell-cell adhesion. I presume you can perturb the makeup of the intercellular space once you know what.
So you can control the expression and efficacy of adhesions, for example.
Yeah. So we can overexpress things. We can knock out things. We don't have genetic lines like other model systems, but we can inject all kinds of things in these cells, and we can inject them in one of the 4 cells. So you can create, so you can do all of those
kinds of things that one can do with quinoflagellates, for example, to understand the different kinds of colony shapes, or is it?
I think we can do more because quinoflagellate cannot be injected, but they have genetic tools, right? So we can do some more spatially controlled things. But yeah, so we can overexpress mRNA. So you can express dominant, negative, or consistently active forms of proteins. You can do CRISPR knockouts. You can do all of those things.
As people try to transfer a plasmid from one species to another one to see what weird stuff.
Yes, this has been done in the context of skeleton development in the sea urchin. So the skeleton is, we know that it comes from the micromeres, that are this small set of cells that you can take from one embryo and put in another. But then the shape of the skeleton depends on the species. So the branching of the larval skeleton, it's species specific. So people have done these sort of experiments in that sense. I have tried to do some of that and mix cells from different species into one another with mixed success, I want to say. Okay, and the last thing I want to just say is that all of these sort of experiments that I showed you in the sea stars can be done with other echinoderms that show this variation in the shapes of the early embryos. And so this is a movie of a sea urchin embryo. This is Litocynus pictus for the ones of you that are interested. And you can see how we can do the same sort of imaging and understand the dynamics of this early embryo shaping. This, just for kicks, it's a sea cucumber, right? Which I was showing before, is going through a whole different set of issues, right? The cells are obviously interacting with the envelope and with the matrix in a very different way than the sea star and the sea urchin. But in the end, you get a very similar shape, right? Yeah.
So it's this irregular shape.
Yeah. So this is the very first video ever taken of a sea cucumber developing. So we are kind of, yeah, I have no idea what it is. So I think just by looking at it, what you can see is this is the same embryo, right? And so the same species. You see how this envelope, it's very different. And I think the material, there is more extracellular matrix-like material in the interstitial fluid. Yeah, I think that's what it is. And they are spreading onto it because they're not just, floating into nothingness like some of the sister species look like. Yeah. So I'm going to play this once more because it's very pretty. But yeah, so I think what you're seeing is that as the cell cycle progresses, they round out for the cell division, and then when they relax, they kind of really make these protrusions into the matrix.
That's good to make the corner.
Yeah, isn't that cool?
But now, do they have like multiple folds to clothes?
Yeah. Same thing. You see them here, actually.
Yeah.
Right here, there. And in the sisters, it's the same. No. In the sisters, there are multiple as well. It's just that normally there is a larger one on the animal side, and on the vegetal side, there are two large holes, and then there are smaller ones on the lateral in the sisters. But there's definitely multiple. And they close earlier laterally, ventrally, and then animally. So it's kind of. All right. How much time do we have? 15 minutes. Okay, so basically, what I wanted to bring home is that these systems of echinoderm embryos is I think really great to try and understand what are the causes of variation in shape, and eventually will give us a handle of what are the effects of variation in shape. And so this is the other type of things that we are kind of doing and working towards. It's using, sorry, I had a exit in case I was out of time. So how do you establish what are the effects of variation, right? And so one thing that we have started thinking about is to simply look at survival and do the survival curves for normal embryos. So we follow the embryos up to to this larval stage, which is when gastrulation is finished, right? We consider that as our endpoint for this analysis. And we simply count, how many of these embryos actually make it to the end of development and have a successful development. And so the control here you see in blue, right? The ones with the small envelope that are more compact have actually better survival than the ones with large envelopes that are less compact. And so that gives us just the big picture. There is some effect of this variation.
Small and large compared to
controls. So these are the same experiments of the calcium of the calcium and magnesium concentrations that make larger envelopes and smaller envelopes. So these are our experimental setting right here.
Does it matter how long you exposed it to those calcium?
So we have done really, really short exposures only, always 2 minutes, and then put them back in regular seawater.
What if you had 2 minutes, but instead of 2 minutes continuously, you had 30 seconds off, 30 seconds on, and gradually change the relative magnitude to see whether this is essentially working like a low pass or a high pass filter.
Yeah, that would be interesting. Yes, I think it can be done. I mean, you mean just to create different sizes or
And or whether there's a memory
of that
of the
treatment.
of the treatment.
Okay.
Because you sort of having 2 continuous minutes, you can spread out that 2 minutes over 10, and then maybe it doesn't remember. And
Yeah. So I think, yes, I think that's a good set of experiments to do. My thinking is that probably because this whole fertilization membrane is formed in about 10 minutes, it's done, right? The more you dilute the treatment over time, the less is going to be effective. But it might have other effects, and there might be some other things. So I think we should do that. Yeah, that's a good idea.
So is this metric also used, so let's say, in this business of IVF? Is
Yeah,
this a similar
it's the
metric?
same thing, yes. So these are survival curves that people do for whatever kind of disease, drug treatment and whatever, that's the framework, right? Yeah. IVF generally stops at, do I have an embryo that is viable to implant? And then that is where you can get really granular information of where development goes wrong. And then after implantation, they just look at the number of successful births, basically.
I was reading in the paper, they have grades, apparently, depending on, I don't know what,
So
maybe it's something like this.
yeah, so the grades that they give to the embryos is based on the shape, actually. So how the embryo looks like, and you choose then the ones that look the best in order to implant. One of the parameters is compaction. Right? If compaction doesn't happen timely or well enough, then the embryo is considered non-viable. And there are experiments that show that it is actually less likely to make it if compaction is not working well. Right? Okay. Yeah.
My own education, the envelope will stay with the developing embryo forever.
Nope. Here, the blastula stage, they hatch out of the envelope, right? Which is something I was gonna touch on, because the fact that you get these drops in survival later on means also that there is a delayed effect, that something is happening at these early stages that then carries on to later stages.
Is that true? Like, giving your data, it seems like even the small large eventually by the later stage it complete drop.
Say it again, sorry.
Like the later stage,
Yeah, it's pretty down, right? Yeah, so at each of these stages, there is a percentage of embryos that don't make it through that stage. And then eventually one of the interesting things to also take away is that even the wild type have not that amazing survival, right? So we are 50%. So talking about robustness of development and all of those things, there is difference of how robust and reproducible development is from species to species as well. There is also, a seasonality aspect of this. We find that in certain seasons, the developmental success in the conditions that we keep them in the lab is higher. We have 80% success. Sometimes we have 40% success, right? So that is, a variable thing per se. And then we can move this percentage up or down based on our treatment of making envelopes bigger and larger, smaller and larger. Yeah.
Are you surprised you found a condition that survives better than
Yes,
type?
exactly.
That's not a loaded question anyway. Yeah.
Yeah, I thought, and especially because of how they look like, I thought they would die because they look all wrinkled, but they actually make it better. And so this is surprising because, well, there are many, this is a whole thing about survival in the wild, survival in the lab. Are we capturing these embryos in their optimal condition, really, or this is what we think is their optimal condition? I don't know, but also, what this means is that if we want to think that this is actually true, the way we culture them in the lab is representative of their environment. If there is a condition like this that can increase survival, it's either the population will eventually go towards that, or there is something else that is preventing it from happening, right? That there is some other reason why having a small envelope is bad for you that we are not capturing here.
To that point, have you ever tried just letting them extend longer in
We
life
are-
to see just what happens?
No, the short answer is not yet, let's say, yeah.
Can I ask a more nuanced question associated with if you have more compacted embryos, does the survival curve potentially might be better, but growth itself is slowed down or sped up? What happens?
Yeah, so there is no growth up to this point, right? Because it's just using the material that you have.
I don't mean maybe the number of cells.
Yeah, don't know. Yeah, I don't. So up to the stages that we can image, there is no difference in the number of cells. But then after they hatch, we haven't really checked.
We followed their morphologies up to the gastrate, stuff like that, and there's no defects.
We are working on that right now. So basically the idea is that now with this system, we can try and figure out what exactly is happening, that change in the compaction is affecting development. And one of the things that we are thinking about is that there is a lot of signaling that is happening already at the blastula stages that starts the cell fate differentiation, right? And in these animals, we know that Wnt beta-catenin is necessary and sufficient to induce mesoderm and endoderm fates that are then necessary for gastrulation to occur, right? And you know, in this system, what we know is that there is nuclear localization of beta-catenin in the precursors of mesoderm and endoderm, and high levels of nuclear beta-catenin induce mesoderm, lower levels induce endoderm, no nuclear beta-catenin ectoderm, right? So we've got this. You know, I don't have to say it here, but like Wnt beta-catenin is one of those signaling pathways that can be affected by all kinds of things. Morphogenesis, right? Cell density, mechanotransduction, and all of those things. So it's possible that we are doing something to that. And the way we are approaching this is that we are imaging nuclear beta-catenin dynamics in the embryos, and we are trying to find ways to quantify these dynamics and see how it is changing in embryos that are more compacted and less compacted, right? So here you're just seeing one of the sister embryos that is now expressing a tagged version of beta-catenin, and we can see, you know, we can see it go in the nucleus in the field of mesoderm and endoderm precursors. And
it's possible you have data, very nice, the compression. I keep thinking symmetry breaking, compression cancellation. You have data to show that under compression and the cells will proliferate eventually, almost like there's a dent. They start to. That reminds me gastrulation. Yeah. And whether
that is connected.
That's connected due to symmetry or due to confinement, pure geometry confinement.
Well, so the short answer again, I don't know yet, but there is a thing. Once they hatch out of the blastula, they build hydrostatic pressure inside, and then they smoothen out, and then they gastrulate. Yeah, yeah. So. Don't know.
Whether you can speed up by confinement.
It might. So Yana is now quantifying the timing. Like while she's doing the survival curve, she's also looking at timing, and the timing does not seem enormously changed. So they develop more or less at the same time on average, right? So what we want to do right now is to try and get these embryos one by one and really look at the dynamic of development based on different levels of compaction, and if we can get some handle on what's going on. Okay, I think I have a couple minutes. Well, let me just finish with this. Like we can, like extreme examples, right? And this is mostly because this is what I have done so far, and we haven't gotten to do the movies in the more compact or less compact sister embryos, but the sister embryo, which is very loose, right? Looks like that. Sea urchin embryo, which is a very compact embryo, right? The pattern of beta-catenin is very different. And so we can sort of start thinking about how is the compaction affecting patterning of signaling and how variation in compaction might affect the differentiation of cells during this process. And that is how I am gonna leave it, I think. So this is the lab for now. We are at the Hopkins Marine Station in Monterey, California. It's a beautiful place, and we are a relatively small lab still, but people are very fun to work with, and we have lots of stuff to do. So if you're interested. Thank you.